Hi all you sake lovers out there, and thanks for tuning in to another episode of Sake Science. This is my third and last video in a series on how amino acids affect the sensory qualities of sake. And in my first two videos, I looked at how they affect the taste and the aroma, but not all of these effects are positive or desirable. So in this last video, join me as I take a closer look at how amino acids can contribute to some of sake's off notes. So actually the first compound I want to take a look at today does not have its own distinctive taste or aroma, but it's important from a regulatory perspective. And that compound is called ethyl carbamate. This compound is actually present in all alcoholic beverages because of basics of yeast metabolism and chemistry. You see, while yeast is fermenting, it takes in the amino acid arginine and breaks it down into urea and ornithine. And then that urea can go on to react with ethanol to form ethyl carbamate. Now, the reason why this is a regulatory issue is that ethyl carbamate is designated a class 2 carcinogen, meaning that it's suspected of being a carcinogen based on animal studies. But before you go and pour out all of your sake, remember that ethanol is a class 1 carcinogen, meaning that it's a known human carcinogen. So, the risk here is not really from ethyl carbamate, but some countries and territories have started to regulate the amount of ethyl carbamate in alcoholic beverages. And one of those countries is Canada, and we can see here the chart of the allowed maximum limit of ethyl carbamate in various alcoholic beverages. And you can see that sake is actually kind of towards the top of that upper limit at 200 micrograms per liter. Now, producers are aware of this issue, and so most of the sake that you buy nowadays is going to be under that limit. But one issue can happen with aged sake because the amount of ethyl carbamate can increase over time. And we can see here in a graph of the amount of ethyl carbamate found in around 30 different sake that has been aged for more than 10 years in the 100-year aging project that's happening here in Japan. And the data points on here are at the bottom is the minimum level, the minimum measurement of ethyl carbamate, the average measurement, and the maximum measurement. And we can see that at the 0-year point, all of the sake measured was under that 200 micrograms per liter Canadian limit for ethyl carbamate. But if we look at the 10-year sake, 10-year aged sake, we can see that that average value is very close to the Canadian limit, and the maximum value is well above it. So it's kind of something to be aware of for producers and importers because it is something that I know some companies and some importers have had issues with, especially in Canada. Now, like I said, producers are aware of this problem, and they've tried various ways of reducing the amount of urea that's produced, because if we can reduce the amount of urea, then there's not any around to react with the ethanol and form the ethyl carbamate, right? So they've tried a few things, but the most effective and cost-efficient way of getting lower amounts of urea has been to develop yeast that either have deficiencies in the enzyme that transports arginine into the yeast cell, or the arginase enzyme that breaks it down into the ornithine and urea. And so there have been a number of low urea yeasts developed in recent years. And among the Kyokai yeasts, you can tell which these are by the K-arg nomenclature. And we can see that there are K-arg versions of Kyokai 7, Kyokai 9, Kyokai 10, Kyokai 14, and also many of the regional yeasts, like the Akita yeasts or, you know, the Hiroshima yeasts, for example, Fukushima yeasts. Some of these are also developing low urea versions of their yeasts as well. And these yeasts are capable of reducing urea values by 70 to 80%, which is a really big decrease. So this is something that is maybe, like I said, an issue going forward, and you might see more of these low urea yeasts being used in the future. So I've actually never knowingly had a sake that uses a low urea producing yeast. And I was interested in trying one because I read that some people are concerned that because arginine is a bitter tasting amino acid, and these yeasts lack the ability to metabolize it properly, that more arginine could be left in the sake, perhaps giving it a bitter flavor. So I looked around and I found something really interesting here. So this is a sake from Murai-jozo in Ibaraki, and it's a new brand called Shinjo, and it's made by their young Toji, who's only in his 30s, and it's got a lot going on here, so that's why I needed my glasses to read all of this. So, okay, so the koji is a mixture of Oriza 1061, which has high alpha amylase for to use with hard to digest rice, and also Roots 36, which is high glucoamylase but low peptidase, so it gives us a lot of glucose but low amino acids. And this is actually Sokujo style moto, but it uses lactic acid that is fermented from Ibaraki lactic acid bacteria, so a native Ibaraki lactic acid bacteria. And the yeast that it uses is that Kyokai K-arg 701. And the rice is Hatan Nishiki, only polished to 80%. So there's a lot of stuff going on here. And then the SMV is -9, so really sweet. The acidity is 2.6, so pretty, acidic there. And the amino acid level is only 0.8. And then the kasubuai is 46%, which is really high, especially for a junmai. And this is a junmai namagenshu. And that 46% kasubuai is like a daiginjo level, with like an 80% polishing. There's just so much going on here that I'm not sure that we're going to be able to tell what effect that low urea yeast is actually having here. It's got a little bit of color to it, just slightly. And wow. So it doesn't have any of those big ginjo aromas, which is something that we would expect from it only being polished to 80%. It's got kind of this light grapefruity, citrusy kind of aroma. Mm. Wow. I mean, it's sweet, but that acidity hits right in the beginning, and grapefruit, citrusy, yokan. It's a kind of citrus fruit in Japan. Mikan, just really juicy, citrusy, delicious. easy drinking if you aren't concerned about the acidity, because it does stand out. I mean, the sweetness is not overwhelming, and it's got a very clean finish because of that acidity. Um, and not really any extraordinary bitterness that I can tell. So, um, kind of a very interesting uh, brand, and I think the Toji is doing very interesting things. So, check it out if you can find it. So, the second compound I'd like to talk about today is called isovaleraldehyde. And this is a compound that's particularly important in unpasteurized namazake, and it can have an aroma that's said to resemble malt or peanuts or other nuts. And the reason why this is an issue in unpasteurized sake, namazake in particular, is because it's part of a undesirable aroma called namahine or mureka. And this arises when we have that malty smell of the isovaleraldehyde and kind of the green herbaceous aroma of acetaldehyde and a little bit of the caramel kind of savory aroma of sotalone, all mixed together. And it's an undesirable aroma that arises from improper storage of namazake. Now, the way that this aldehyde is formed is actually from isoamyl alcohol being acted upon by a surprisingly very specific enzyme from koji that oxidizes that isoamyl alcohol. So it's an isoamyl alcohol oxidase. Now, this aldehyde can actually go on to react with oxygen to form isovaleric acid. And this is a compound that contributes to the smell of natto and sweat and also stinky cheeses. So it's not something that you think we necessarily want to have around in sake. Now, as I said, this is one of the reasons for keeping namazake chilled. But what's interesting is that this enzyme can still be active even at pretty low temperatures. So this is an example of one sake that was stored at 3 different temperatures: 30 degrees, 15 degrees, and 4 degrees C. And we can see that a lot of isovaleraldehyde was formed when it was warm, but there's also a significant amount of it formed even at 4 degrees Celsius. It's over the detection limit. So this is kind of an issue for all namazake, no matter how you store it. And researchers have looked at lots of different sake to see if there's a certain type or a certain specs that lead to more production of this isovaleraldehyde. And the only thing that they really found that was consistent was when there was a higher total protein level, there was a higher production of this isovaleraldehyde. And so that high total protein actually represents more presence of that enzyme from the koji. So the thing that we want to do is get rid of that enzyme, right? And there are a couple ways to do this. One is through pasteurization, which will denature the enzyme. The other thing that can be done is ultrafiltration, which is an expensive step that will actually remove the proteins from the sake so that there isn't that enzyme around. And finally, another thing that can be done is using koji that has less of that enzyme to begin with. And one example is this namazake senyo koji from Akita Konno, which produces less of that enzyme. So we can definitely get rid of this off note, but something I like to think about is that, it's kind of a natural part of sake when we think about it. And whether something is considered an off note really depends on concentration, context, and intention. And there are definitely people who are experimenting with aging namazake specifically to get this aroma. So one thing that you can do is actually try this at home. So I did this, and I stored a namazake. I'm not going to show you what it is because I don't want to reflect badly on the producer, but I wanted to see for myself what this actually smelled like and tasted like. This was stored for a couple of weeks at temperatures between 15 and 20 degrees Celsius, kind of in a cooler part of my house. The color here, let's see, there isn't a lot of color. And the aroma, now when I had this, the reason why I wanted to do this on my own is that I've had aged namazake before, but I didn't know what it smelled like before it was aged, right? And so this one, when I had it before, it was really bright, really fruity aromas. Um, and this is not bad. It's actually not bad. It smells more savory. It doesn't necessarily smell sweaty. I think I can get what people are talking about with that namahine because I get the acetaldehyde and I get a little bit of the sotalone in there. It's actually reminding me of kind of like apple pie, like cooked apple and pastry. And now I'm wondering because I've had that or I've perceived pastry kind of smell in other things, and I'm wondering, is that isovaleraldehyde that I've been perceiving all this time? That would be very interesting. It's good. It's good. Now, I don't know. This is a very high alcohol. It's 19% Genshu. Um, what I remember from drinking it before I aged it was that it was very bright and kind of fresh. And this is just kind of mellowed out a little bit. Again, kind of like cooked fruit, apple butter, apple jam, pear butter is what I'm getting. Um, Really interesting, and I kind of like it. So again, even though isovaleraldehyde is considered an off note, if it's part of your intention to create it, and also if you're aware of the context, it's not something that was just uh, handled haphazardly at the brewery, and this was done for a specific purpose, then I think this is totally acceptable and very interesting, actually. So the next topic I'd like to talk about is Strecker degradation. And this is one of those Maillard reactions that can happen as sake ages. And basically this is a chemical reaction where sugars break down into reactive compounds called dicarbonyls. And then these dicarbonyls can react with amino acids in a very complicated series of steps to give us the what's called the Strecker aldehyde of that amino acid. And a few of the more important aldehydes that are formed are the first is isovaleraldehyde. So there are actually two ways that this can form in sake, making it seem like, this is actually a compound that we can expect to be around in sake. And this is formed from leucine. And then there's benzaldehyde, which forms after a series of reactions from phenylalanine. And this has the aroma of almonds. And finally, there's methional, which is formed from the amino acid methionine. And this has an aroma that is characteristic of cooked potatoes and like French fries and potato chips. Methional is really one important compound because as a sulfur compound, very small amounts of it can have an outsized effect on the aroma of the aged sake. And we can see here a graph of how methionine changes in sake that has been stored at ambient temperature and at cooler temperatures under 15 degrees C. And we can see that the um, the odor activity value is pretty high even after aging for 3 years in the ambient aged sake. And I don't know why that data point goes way down at the longer aging. I think that might be an artifact, and there was actually, these are average values, and there was actually quite a big range. But we can clearly see that even the cooler aged sake has average methionine values increasing the more that it's aged. Now, in the past, it was thought that methionine was also directly the source of even more undesirable compounds called polysulfides, such as DMDS and DMTS. DMDS is said to have the aroma of garlic or also the seaside. And DMTS is said to have the aroma of uh, like nuka or pickled vegetables, but also rotten vegetables like rotten cabbage in very large amounts. So these are compounds that are really considered undesirable for the most part. But it was found that methionine does not directly form these compounds. What actually happens is there's a cycle of methionine in the yeast cell, and the yeast uses methionine to form polyamines that it needs for various metabolic purposes. And in the process of regenerating the methionine, it produces this intermediate which can go on to form a precursor to these polysulfides called DMTSP1. So the DMTS precursor. And it's this precursor that breaks down to form those polysulfides. So researchers are really interested in the conditions that lead to more formation of DMTS, and some of those are higher rice sulfur content, basically meaning more content of those sulfur-containing amino acids, such as methionine, more yeast death and lysis in the moromi fermentation stage, and also higher pressing pressures, especially in the seme portion of pressing. And this is another issue where yeast breeding has been useful. And just a few years back, a Kyokai yeast was released that has a defective methionine regeneration pathway, and so doesn't produce that intermediate that produces the precursor to DMTS. And that yeast is called Kyokai MDED1. So especially for producers who want to limit the amount of DMTS that's formed in shipping and things like that, this is a yeast that they might be interested in. But again, the formation of DMTS is something that happens naturally in sake. And what's interesting is that just the presence of DMTS is not considered negative. What's important, again, is the balance between the different aromas in the sake. So when we have a sake that has a lot of sulfur compounds, but not a lot of those other aged aromas such as sotalone or the Strecker aldehydes or the ethyl esters of organic acids, that's considered by sensory evaluators to be hine aroma, which is the old undesirable aroma. But when there are those other aged aromas present, it's actually just considered part of the overall kind of aged aroma of sake. So again, it's all about balance, context, and intent when it comes to these off notes. So the final thing I want to look at today is the effect that light, especially UV radiation, has on sake. And somebody asked me online, is the light strike that happens in sake the same as what happens in beer when it gets skunked by light? And the answer is surprisingly no, they're completely different processes. You see, in beer, there are these compounds called isohumulones, which are derived from hops. And these are sensitive to UV light and also sensitive to degradation um, with visible light when it excites flavins, especially riboflavin, in the beer. And that can lead to the degradation of these isohumulones. And the same thing can happen with the amino acid cysteine. So it can break down from UV exposure and also visible light exposure and reaction with those excited flavins. And this forms sulfhydryl radicals. So then the degradation product from the isohumulone and the sulfhydryl radical from cysteine can react to form MBT, which is the specific compound that gives beer its skunky aroma. But sake obviously doesn't have those isohumulones from hops, and so the light strike that happens in sake is caused by UV radiation causing the breakdown and reaction of the amino acids tryptophan to form indoles, especially this indole called scatol, which can smell like jasmine at low amounts, but it's also a characteristic component of feces. And also compounds called, what are they called? Carbolines, when the degradation products react with sugars in sake, and these give rise to characteristic colored compounds. And then the other amino acid that can react with UV light is again methionine, our old friend, and this can break down to form a methyl mercaptan, which is the characteristic aroma that they use in, they use mercaptans in natural gas to give it its odor. So it has the odor of rotten eggs or farts, basically. So researchers know that it's UV light under 400 nanometer wavelength light that is the cause of this light strike in sake because of experiments that they've done using different filters from light sources. So this is an experiment where they used a strong halogen light source where it was the sake was shielded from the heat from the light, and they used different filters to see how the sake was affected. And they used the change in color, so the change in absorbance of the sake, to see as a marker of how the sake was affected. And so we can see when there was no filter used, so all of the UV light was coming through, that there was a large change in the color of the sake. And when they used a blue filter that allowed some of the ultraviolet light to come through, there was still a change in the absorbance, but a little bit less. And when they used green or red filters that completely cut out the ultraviolet spectrum of light but allowed visible light to come in, there was basically no change in the absorbance of the sake. So the lesson here is that light strike can be prevented by, well, number one, keeping your sake out of bright uh, light, especially sunlight, and second, by storing it, bottling it in darker colored bottles. And again, I have a sake that I prepared here at home I'm not going to show you what it is again because I don't want this to reflect badly on the producer. And this was a sake in a light blue bottle. And I stored it for about 2 weeks in bright but not direct sunlight at a south window. And I could tell there was a change in the color and the aroma of the sake even after a day. And we can see, I think you can see there, it's got this kind of, it was originally very clear crystalline color, and now it's got this kind of, I don't know, kind of dirty yellow color. Ooh, ooh, yeah, that's bad. Um, some people have said that that light strike in sake can smell like wet cardboard. I'm getting dog. This is like wet dog to me. Maybe kind of that barnyard smell. I actually don't want to put this in my mouth. I would. It's not super strong, but I don't recommend storing your sake in bright sunlight. So, how was that? Was that interesting? Did I help you understand the role that amino acids have in forming some of these off notes in sake? I hope so, and I hope that this can help you, for example, think about how to store your sake properly, how to recognize some of these off notes in sake that you buy at the store, and also to maybe change your perception of some of these off notes in the way that they're not always completely unacceptable. Again, I want to go back to my mantra that I've been saying about off notes recently, which is balance, context, and intention. So even though something has been aged and has some isovaleraldehyde, for example, or maybe it has some DMTS in combination with other aged aromas, those can sometimes, give sake its complexity. And so even though there's been this kind of push in recent years to make these really clean sake and develop koji and yeast that don't produce any of these off notes, I think that there is room for both. Sometimes we don't want those off notes, and sometimes we can accept them. And what's important is being able to understand them and recognize them. So if this was helpful for you, please smash the like button and subscribe, and stay tuned in the future for more episodes from Sake Science. And until then, keep exploring sake.